good morning, good afternoon. It's my pleasure to welcome you to this EAHAD webinar on behalf of EAHAD, on behalf of my colleagues today and the speakers. This is a webinar about the hub and spoke model, and it's from EAHAD. So the European Association for Haemophilia and Allied Disorders is a multidisciplinary association of healthcare professionals caring for people with hemophilia and other bleeding disorders. And its members include hematologists, internists, pediatricians, nurses, physiotherapists, laboratory scientists, and researchers from all over Europe. And in EAHAD, there are several very active working groups. One of them is a working group on gene therapy, working on several projects. Now, gene therapy of hemophilia. There's a lot of progress, and there are some recently published data on the efficacy and safety from phase 3 clinical trials. But we know that the preparation, administration, and follow-up are completely different from other hemophilia therapies. Currently, the application for approval has been validated and being reviewed by the EMA for both hemophilia A and hemophilia B. Therefore, it now can be discussed how to prepare the care structure if gene therapy is approved for hemophilia. Next slide, please. And for this reason, it was in 2020, a hub and spoke model was first proposed by Flora Peyvandi and Declan Noone, and this was promoted for the care of hemophilia and rare bleeding disorders with gene therapy. Next slide, please. At the same time, we learned more and more about the challenges for hemophilia centers. Some of these challenges are listed here and described in a paper published in the journal Hemophilia. Next slide, please. And this was a joint publication of EAHAD and EHC describing different models of collaboration between a hub center, so a hemophilia treatment center performing gene therapy, and different spoke centers, which are also hemophilia centers and taking care for the follow-up of the patients. Next slide, please. And the different responsibilities and the cooperation between the centers are a complicated process, and recently it has been added that it would be important that the dosing centers remain hemophilia centers, and the hub and of course also the spoke centers are familiar with all the hemophilia related issues and also can provide competent care to patients for all hemophilia related issues. Next slide, please. Now we can look forward to 4 lectures we have today from our very distinguished speakers, who all are members of the gene therapy working group of EAHAD, and they will highlight different aspects of gene therapy. The first one will be given by Hart from UK with a very up-to-date overview of the recent results, including those presented during the last congresses. Then Robert Klamroth, who is president of EAHAD, will give an overview of which patients have been included in the gene therapy trial so far, what are the inclusion and what are the exclusion criteria, and what is the role about the anti-AAV antibodies. And this will be followed by 2 presentations with the requirements, but perhaps also the pitfalls of gene therapy. One from the patient's point of view given by Declan Noone. Declan Noone is the president of the EAHC. And one from the doctor's point of view. This will be given by Flora Peyvandi. Flora Peyvandi is a past president of EAHAD, and it's really a great pleasure for us that we have Flora Peyvandi together with us during this webinar. So please take the opportunity to ask questions. The presentations will be each about 10 minutes, and I hope we will have enough time to answer all the questions you may have. So with this, I would like to ask Dan Hart to start his presentation.
Thank you very much, Wolfgang, and thank you very much all for joining this EAHAD webinar. And so 10 minutes is a short period of time to give you an overview about gene therapy in haemophilia, recognizing this is a very fluid area. And I'll summarize at the end about some initiatives I think that will try and keep us all up to date, some from the EAHAD and some from EHC, to enable us as interested clinicians and MDT members, as well as patient advocates, to really help keep our community and ourselves informed. Next slide. These are my disclosures. Next. So we need to always be clear what we're talking about when we're talking about gene therapy. And as a slide that I used to use a little while ago, it was always on the left. So next for the animation for the gene therapy. Next slide. To show that it's in the red box, this has been what really we're concentrating on today, which is about a new gene being transferred into the cellular nucleus without any removal or modification of its existing DNA, so an episomal strategy. I think what's interesting, having used this slide over a number of years, is now actually that cellular therapy and gene editing is emerging into trials at an early phase, and we need to keep our eye on these different strategies. But for the sake of today, we're going to be largely concentrating on gene therapy as defined by the red box kind of strategy. Next slide. And many on the call will be fully aware of this, but in case there aren't, our objective is essentially to give a single treatment by a simple peripheral infusion as an outpatient that ultimately has the ability to achieve a durable and meaningful factor expression for years and hopefully decades to that individual. And that's about a packaging within a viral capsid, and that having a liver tropic delivery system in which that AAV viral capsid is able to deliver the gene of interest to the hepatocyte, and subsequently for the first time in that patient's life they can actually produce and then secrete the protein 8 or 9. Next slide. And actually, we've learned through the evolution over decades, and this has been a long journey to get to this point in our specialty, that adenovirus, for example, on the left was not the right way forward because of the immunogenicity issues, albeit it had a larger genome option. And it was on the right, the adeno-associated virus, that had a lower immunogenicity and a poor genomic integration, which is to its advantage that we're not looking to integrate this gene into the chromosomal structures of the hepatocytes. And just keep your eye on the middle group there, the retro lentivirus, where there is a genomic integration strategy that is beyond the remit of today, but I think there are strategies in the background to utilize this as a strategy as well. So I think we always have to be mindful of what's present for us, but what also is coming up. Next slide. And thinking about that, then an adeno-associated virus, next slide for the animation, to see that actually this is called a clade. So it's the clades of evolutionary change in this virus. And you can see in the pink cloud, so that the serotypes that are being used in different platforms of gene therapy, both for haemophilia A and B. I think it's quite interesting. There's a discrete difference between maybe the AAV5 and some of the others in the other groups. So how we as clinicians understand that, are there differences or not between these different serotypes, is I think a question that's open, and we need to continue to try and understand depending on the immunogenicity of trial data and then subsequently in the real world data. Next slide. And I think to remind ourselves that actually the wild type AAV that has a replicative capacity, that's been stripped out on this next slide. So we've got essentially the chosen gene cassette with either the factor 8 or factor 9 gene internally that's then able to deliver the gene, but clearly there isn't a replicative capacity beyond that, which is important, I think, for our patients to be aware of. Next slide. And I think this were the editorials of nearly 5 years ago, and gene therapy has always been in touching distance, hasn't it, for haemophilia? And I think the cure being within reach, albeit that no one really likes to use the C word for haemophilia, there clearly is data out there that is tantalizingly close to a cure, albeit there's variability across a number of platforms. But there clearly are some individuals who are achieving both a functional and a pragmatic cure, as defined by kind of avoidance of prophylaxis or indeed just normalization of plasma measurable protein. Next slide. But I think we need to be aware that there's multiple platforms, and the runners and riders in the gene therapy race is very important for us not to be distracted just by the front runners. Next slide. I think that the race is clearly on, and there's clearly a commercial slant to this, but for us on the clinical side and our teams just understanding what the implications are for patients who can make a single choice to opt into an AAV-based platform, and getting the right treatment at the right time for that individual is part of this kind of hub and spoke model that gives ample time for them to really understand what their options are, both in terms of non-gene therapy versus gene therapy, but within the gene therapy platforms to understand what the pros and cons might be. Next slide. So I'd urge you all to keep an eye not just on the front runners. It is very important that we have front runners that are testing the regulatory authorities to bring successful data to be scrutinized, and then hopefully then gaining regulatory approval. But I think we also need to be mindful of what's going on in the pack in some of those following along that may be close to final phase and pivotal studies, or indeed early phase, that may be paradigm shifting downstream, that may start to influence whether a patient decides to go ahead with gene therapy next year, or maybe waiting for a number of years before going ahead with such a strategy for themselves. Next slide. So it's beyond the remit of my talk to really go through the data in any detail, but just indicative of the number of different platforms out there, that many of these will be familiar to you on the call, and there's variable kind of mature data for these that is worth looking at, and there's clearly some that have been successful through to pivotal phase 3, others that have got moments of pause, and I think it's just keeping an eye on all of these as we go. Next slide. And I think I use the arguably the most mature data for the BMN 270, the BioMarin valoxacogene roxaparvovec molecule, to demonstrate what we should be looking for in data as it matures, but also how it's presented to us. And you can see these box and whisker plots from left to right across the x-axis is a timescale over to 4 years, and then a continuous non-logarithmic scale on the y-axis of factor VIII activity as measured by a chromogenic assay. And so already we've scrutinized the axes, and we can see there's both mean and medians within the green boxes, and we can see there's the whiskers, the lines that protrude either side, that are from quite low levels at all time points to quite high levels, and certainly during the first year above the top end of normal. There's quite specific allusion to this being chromogenic rather than one stage, and you can see at the bottom that is included, that the one stage reads higher than the chromogenic, and you can see that then there's both mean and medians within the table at the bottom. So without scrutinizing BioMarin in isolation, you can see that there's a lot in this table and this figure, and this is the kind of data we need to be seeing in a consistent way across platforms so that we can make judgments about performance as year-on-year data matures. Next slide. So haemophilia B similarly has a range of platforms coming through, and most of you will be aware the Padua variant, the gain of function variant, a single point polymorphism identified in 2009 by Simeone, has been utilized to great effect. Next slide. Given the amount of protein that is actually expressed, you can see here on the right-hand side the tall, darker green column. For the same amount of pale low column protein expression, there's a lot of activity, and this ability to leverage this gain of function in gene therapy has been a huge advantage in the haemophilia B setting. Next slide. And as such, you can see the first two rows in the early phase wild type factor 9 gene therapy trials, although they're single figure expression, there's important durability data coming from these trials that is giving us, I think, a lot of reassurance on the haemophilia B setting of durability year on year. And then the conversion to Padua variant use in the bottom two rows for the Pfizer and the Unicure, that there's close to normal activity levels, albeit not so long follow-up as the phase 1, but it's the equivalent amount of protein being expressed, and it's that gain of function which is giving the leverage in the activity. So I think there's a great optimism about durability over years for the haemophilia B setting. Next slide. And so really to finish off, I think the hub and spoke discussion that we'll have now is then, I think, offering individuals the opportunity to understand whether gene therapy is correct for them, and for both us as on the treating side of the fence and the person who is about to be treated or considering being treated, thinking about key questions to identify differences between gene therapy platforms, which may be about eligibility criteria and dosing through pre-existing AAV immunity, for example, the differences in vector dosing, the risk of liver transaminase rises, the immunosuppression strategies, and that range of expression and durability that I've alluded to. So next slide. And I think we have to recognize that it is increasingly difficult to keep up with all the gene therapy developments. There's a lot of very hard science in there. I think many of us have got used to talking about AAV, and there's multiple platforms, but there's other modalities coming through that we're going to have to learn about too. I would certainly really recommend the excellent periodical from European Haemophilia Consortium, Novel Treatments in Haemophilia, that is a key document in my view for comprehensive review of public domain data on many molecules in the space, and also the emerging repository that Professor Miesbach has really spearheaded to get a team together to curate the gene therapy publications and announcements to support our community. And this discussion about hub and spoke, I think, really gives us an important opportunity to think about how we convert from trial activity to service delivery ahead of the first regulatory approvals that may be as close as end of the year or start of next year. And on that note, with the next slide, I'll leave you with a view from our clinic in London. Many thanks.
Yeah, thank you very much, Dan. So my topic is patient selection and eligibility criteria. Next slide, Angelos. Next slide. So here are my disclosures involving several companies. Next slide. So I would like to start with some general remarks about patient selection for gene therapy. I think in the times nowadays, the patients are at the center of decision-making about the treatment, about gene therapy or other treatments. And if the products are licensed, one point is that we have equal opportunity for our patients to access gene therapy. That might be different in different countries, but in general, there should be for all patients a possibility to have gene therapy. There's a lifelong follow-up to ensure long-term success in these patients. And then I think even for patient selection and eligibility, the hub and spoke model is the best model, because the spoke centers know their patients. They know how they are treated. They know if they are committed. They know if they have the right eligibility criteria. And the hub center knows about gene therapy, knows how to treat these patients, how to treat gene therapy. And the close collaboration, even in the phase of patient selection, is from my point of view important. Next slide. For the patient selection, first of all, I think information about gene therapy should be applied to all patients, nevertheless, if they're eligible or not, just for information. And that's what we are doing in Germany already, because you all know 2 products are, one in hemophilia B, one in hemophilia A, are applied for the EMA. And I guess in Germany, maybe in the end of this year, beginning of the next year, the products will be on the market. And first of all, for the selection of patients, the clinical trial inclusion criteria will be the initial template for selection. And then Hart already mentioned something during the study. So we are talking about patients with severe hemophilia A or B, at least 18 years old. There is, in most studies, no pre-existing antibodies against AAV, at least for all studies in hemophilia B and for all hemophilia A, and all studies in hemophilia B except one. And liver health is an important topic, now active infection, and now HIV in most studies, not in all studies, and now inhibitors. I will focus a little bit more on this medical criteria, but that is not the only point. There are other criteria too, because we all know from the clinical studies, and that will be the same when the product is licensed, there are certain challenges for the patients. So, of course, it has to, the controls, and Flora Peyvandi and Declan Noone will reflect more on this, what commitment of patients is necessary, but of course, for the selection of patients, you have to take it account in advance. What is the family planning? What is alcohol use? What is motivation and commitment? If you first look in the next slide on medical eligibility, so I think the biggest hurdle in the beginning is the neutralizing antibodies against AAV. Then Hart already mentioned that we have different serotypes of AAV. And one difference is, of course, that the seroprevalence of neutralizing antibodies against different types of AAV are different. So depending on the serotype, the lowest antibody titers we have in AAV5, and there are differences between countries and age. And we can expect, if we look across Europe, there will be neutralizing antibodies against AAV from 20% up to 60%, depending on country and age and serotype of AAV. And for all studies in hemophilia A, a negative test is required. So patients with antibodies against the viral vector, which will be for the first licensed product AAV5, cannot be included. For hemophilia B patients, there's at least one study, and that will be the first licensed product, where patients with a low and moderate antibody titer had successful gene therapy with sufficient or good factor IX expression. And from this study, an antibody titer up to 1 to 700 will be allowed for inclusion in these patients. Only patients with very high titers of AAV5 neutralizing antibodies will be not suitable for this kind. But that's only one study. All other ongoing studies in the field of hemophilia B are excluding patients with neutralizing antibodies. So that is the first step for patient eligibility, if there are neutralizing antibodies present or not. The second step is, because Dan already mentioned it, the production of factor VIII and factor IX will be in liver cells and hepatocytes. And so evaluation of liver health before gene therapy is, from my point of view, of importance, because you need a healthy liver to have a successful gene therapy. And you know that a lot of patients had viral hepatitis B and C, and of course there should be no ongoing infection. Successfully treated patients with hepatitis B and C can be included, but of course we need some markers for liver health, and we have blood tests, of course, transaminases, we have the sonography and the elastography to see how well the liver is, because in patients with advanced fibrosis, it's usually an exclusion criteria, and we have a higher risk for complications. And after this step, of course, patients with inhibitors against factor VIII or factor IX were not included in the trial, so we need a negative inhibitor test. And I think what is of importance too, if we look in more the future, are the comorbidities and the potential liver toxic medications, which can play a major role in the first 6 months when we have this regular controls. From the other point of view, in the next slide, we have some general eligibility, and that is managing patient expectations. And I think it's really important, and there the Spoke Center has the first role to introduce to patients gene therapy and the possibilities and the challenges. Always you have to compare to the current treatment of the patients. A lot of patients are doing very well. Of course, you can offer gene therapy, but you have to find the way to explain what it meant to you. At the moment, family planning, at least for 6 months, should be postponed. And the requirement for follow-up should be explained very well. That will be the topic of the next two calls. So from my impression from patients I included in gene therapeutic studies, and that will be pretty much the same when the product is on the market, motivation and commitment of the patient is crucial for successful gene therapy. In my last slide, I want a little bit to summarize. So patient eligibility will be according to the licensed product. That means AAV negative for all hemophilia A patients and AAV negative with low titer or moderate titer for the first licensed product in hemophilia B. There should be a shared decision after discussion of all treatments options with the patient. The first role is with the spoke model, and then the second role is in the hub model. And already with the selection of patients, there should be a close collaboration. And important, very important for patients is commitment for short and long-term follow-up. And that will be the topic of the next two presentations.
Thank you very much, Robert and Dan, for giving such a good overview. So I'm going to talk about the patient journey in terms of gene therapy from a patient point of view. Next slide, please. So we've just heard from Dan and Robert about all of the variations that are coming in the gene therapy space. And we're talking about a hub and spoke where the patient is sort of between the two centers. So how would all of that really work? And that's something that we really need to think about as we move forward. If I go to the spoke, it is my trusted center. I've been with them for a long period of time. Is my home center, usually close to my home, and I have a long-standing relationship. In the hub, I have the expertise in looking for any aspects of evolution of steroid use or any aspects of use of gene therapy, the response rates, and both of those are incredibly important. So how do I ensure that the patient is not caught between the two and possibly falls between the cracks? The next slide, please. So this is an attempt at trying to put the entire conversation that Robert and Dan have illustrated onto the one slide. It is very busy, so I'm going to broadly discuss it. So the initial discussion from a patient perspective, I'm interested in gene therapy. What we need to do at the spoke level is to ensure that we have enough information to give a broad overview. And what we need to do is we need to access simple information for the patients on how do you give that information. Broad strokes of what gene therapy is and not into too much depth at that point. And the main goal is to ensure that we have a broad piece of information before we go any further. As Dan illustrated, there's a number of different strategies. There's a number of different types of gene therapies coming down on the market. So what we are really talking about next is eligibility. There's no point in thinking about 7 different types of gene therapy or even one gene therapy if you're not eligible. So what we need to think about at that eligibility stage is ensuring that we have neutralizing antibodies identified, liver health monitored, cardiac health monitored, so that we have that information. I think the challenge here is probably less to do with the patient and more to do with how that is networked on a national level and making sure that that information is stored appropriately and shared between the hub and the spoke. And the main goal at this point is for the patient is to identify a direction. Which way are we moving? We then have to create this education space, as Dan and Robert illustrated so nicely, talking about family planning, talking about comparisons of treatment, talking about the adverse events and what may or may not happen in relation to additional medications, talking about response levels, talking about activity levels. Ideally, that would happen at the hub, because then you have a large enough patient population to create a peer-to-peer group, or at least a conversation between individuals at the hub. What I would hope and want as a patient is that the clinician at the hub shares that information with the team at the spoke. With the aim of the education challenge is, how do we give the education in the right way to make the decision a go or no go as we move forward. But at this point, we're still on a holding stage. Then we moved into the informed decision concept of, how do we help the individual patient weigh up what's best for them? We need to have a very structured conversation at the hub, with the hope that we have a clear idea on what are the benefits and risks for each individual. Again, that is something that I would really like to be transmitted to my spoke, just so that they're aware of the thinking and what's going on. Our key challenge here is helping the individual weigh up the education and the benefits and risks for them, so that we move forward in that go, no-go concept. In terms of dosing, then we're on the next stage, which is kind of the final stage before the long-term follow-up happens. And what we really need to do is offer the point there for questions for the individual. In the hub, we want, ideally from a patient perspective, I want that in the hub, because what we want to do is make sure that the dose is correct, that all of the eligibility and planning is in order, and again, that information transferred back to my spoke. Ideally, what we're trying to identify with the hub in this respect, from a challenge perspective, is if there are any red flags appearing, they're identified early, and each patient gets their best opportunity at gene therapy, and that's the goal. Then in terms of follow-up, the first year here you can see is quite busy. A lot about giving bloods in the hub. I would hope they would give directions in terms of steroid medication or cardiovascular medication if required. And also what we hear back is that people want to talk to other individuals in the same space, just so that they have that ability to share. And we have very good French programs that we can see as a good idea. As we move forward then into the spoke, and that's where I want to be, where the blood's to be taken, where the treatment to be prescribed, but we need to improve that coordination. Our challenge is, how do we monitor the factor expression and any other additional medications in a key consistent way? And the goal is making sure everything is ticked off the box straight away. In years, follow-up in years 2, I'll wrap up pretty quickly, is broadly monitoring at the hub. I think in the spoke, I think the conversations will possibly be starting to change depending on the response of the individual to the gene therapy. If the factor expression remains high, it may end up with discussions around how do I do, or I want to do more or more strenuous activities, and that's a discussion that needs to happen. Or the expression is coming down, and then facilitating a conversation there is incredibly important on what the expectations are. So the challenge is how to identify the next steps, and the goal is to avoid any additional damage and make sure that the treatment plan going forward is defined. And then follow-up in year 2 in the hub center. Ideally, what we would like is a liver biopsy, if that is something that is required, but general monitoring in the spoke of the liver and the factor expressions. And with the aim, the challenge is, how do we manage this in the system all going forward? And the goal is to increase the person's benefit from the approach of gene therapy. And with that, this is one slide that shows the entire conversation. So we'll be very interested to hear the audience's response to this type of concept in presenting it to the patient organizations. With that, I'll hand over to Flora, and thank you very much for your time.
Thanks very much, Declan, and good afternoon to everyone, and to Wolfgang for organizing this group of speakers. I try to report my thoughts in the last months of gene therapy, and as a physician, what are the idea I could have and how these ideas are changing every day. And based on the new data that are getting to the literature, of course, we are improving our knowledge. And I'm going to share some of the data that I have in my mind, and most of them have been shown. And at the end, we can just come over what is my vision. The next slide, please. So we heard gene therapy is differing compared to other type of treatment because the therapy is just one single intravenous infusion right now with adeno-associated vectors. And this material should get to the target cell, which in this case is treating the liver. And that's the reason why the health of the liver, Robert explained, is very important. And then you have an endogenous production of factor 8 and 9, and it's important to understand for how long time. And the next one. Now, as a physician, there are really several questions, and every day I'm adding one question on top of that. So the first question, now we have a lot of different substitutive and not substitutive therapies for hemophilia A with and without inhibitors, and they are working very well with a high efficacy and safety. So whether the gene therapy is adding more to the benefit of my patient and the personal benefit of my patient, how much is there? But for doing that type of analysis, I really do need to accurately know what is the efficacy and safety of each single instrument, therapeutical instrument that I have in my hand, and how much is good for my patient to change the treatment of him, and whether I'm really changing the quality of life of patient, and how I'm going to and making the measurement of the quality of life. That's a very important question, and which price I'm paying for that, and how much the health system is paying the price for the gene therapy based on the improvement that I have more for my patient, and whether my center is a good center for the treatment based on the hub and center description that has been done. And last but not least, what is the motivation and commitment of my patient, which was explained clearly with Robert. And again, what is the personal benefit for my patient? And next one. So as regards, the first point is, what is the safety for my actually choice? How safe is the gene therapy, and what is the efficacy of different type of instrument which are available and which is going to be in the market soon? So as you've seen, Dan explained, there are 2 clinical trials on the phase 3, and both of them for hemophilia A and hemophilia B would be BioMarine and Unicure, showed clearly, the next slide, that we have a good expression, which is very high at the beginning, and for factor 8, the level is getting normal, and after a year we have a reduction, and this expression is remaining for almost 5 years, and we have expression around 8 to 10% for now, and none of us knows at this point for how long time this expression would remain. For factor 9, the level of expression is about 35 to 40% and is more stable compared to factor 8. And we do not have with this gene variant of Padua, which is making the expression of 40% for a long period of time, more than 2 years, but for the experience that we have with the gene therapy of factor 9, the expression could remain for almost 10 years. So there is a little bit of difference between 8 and 9, and that should be taken in mind. And of course, it's important for how you are evaluating the cost of these products in the future. The next slide is more or less showing it's important to look on factor 8. You can see there is actually the level of expression is reducing after, I would say, after 18 months, after 1 year, but the reduction is remaining a little bit less severe compared to the initial part. And that's a very important information that we have to keep in mind. So after 2 years, we have to see now for how long time this expression is remaining. Next slide is showing what has been shown. There is no doubt that most of the patients who are in phase 3 clinical trial, they had no bleeding, 0 bleeding, and they were more than 80%, and the efficacy is very good. And for factor 9, as I said, the next slide is showing that the level of expression is much more stable, and we need to see how long time this expression is remaining, similar to what we have seen in the first 2 years or 18 months. Next slide is just showing, even for factor 9, there is no doubt that there would be a significant improvement in the annual bleeding reduction, more than 64%. And next slide is just trying to show what about the safety. The most important and relevant point for the physician right now and patient is the elevation of ALT. And right now, we are just trying to understand which level of ALT increase should be treated with a steroid and corticosteroid, because even the treatment of corticosteroid is not a piece of cake and for the patients. And we have to understand for how long time we have to treat our patients, whether we have to treat them upfront and for how long time. The long-term adverse event is the genomic integration. Since now we have not seen actually any kind of tumorigenicity in the patients treated, and I'm going to report the data for you, but we have to keep an eye on that. And we know that AAV could make the integration between 2 to 5%. If you see the next slide, is just showing that there were one subject who developed a hepatocarcinoma in Unicure study, but very clearly has been analyzed and showed by the genetic analysis there is no correlation. And that's the reason why Robert was mentioning the liver health is extremely important, since this patient was one of those that did have the hepatitis and cleared for the hepatitis C. So then at the beginning, we have to be very careful about the selection, not involving those patients that they have important liver diseases. For biomarine clinical trial, in phase 1 and 2, there was one salivary gland cancer, and analyses right now are under the investigation, and we are very much looking forward to see there is no correlation within a gene therapy. And of course, we have to keep an eye on that very clearly for the future. Next, so this is what is the condition with efficacy and safety. But do we have the facility of using this gene therapy when they are getting to the market in 2022 or 2023 for all our patients? I would say no, because we have portion of our patients, they have inhibitor, again, factor 8 and 9. There are portion of the patients, about 50% of them, they have the neutralizing pre-existing antibody against the capsid, and they cannot be involved. We could not involve a pediatric patient because there is a large quantity of AAV that could be lost during the liver growth of the children. And that's what we need to think about the new strategies in the future. And of course, there are different type of clinical trial now for the inhibitor patients. And as we said, those that they have the problem with the liver, and of course, those patients that are not motivated and they do not want to go through the gene therapy with the doubts that we have. So it's very important, the selection of the patients and talking with our patient and involving them in this decision. Next one. Next one is how the quality of life of my patients have been changed and how would be changed. I think there is no doubt if things works well and there is no side effect, there would be a reduction of ABR, there would be one single shot, and this patient, they would have less pain and discomfort, and we would have better quality of life with the more activity. And this data in the next slide, you can see has been shown by Robert and Wolfgang on their patients. However, there is a portion of anxiety in this patient have been clearly reported in this paper. The patients want to know how long time this expression would remain and for how long time they would have the similar type of the result. And that's the reason why we do need to talk with our patients and we need to inform them about where we are standing and how much knowledge we do have. And next slide is just showing the cost. The data that has been reported right now is much higher compared to what we are expending right now for factor 8 and for emicizumab. The cost of factor 8 is changing between 40,000 to 200,000 yearly for our patient, and for emicizumab is between 80,000 to 250,000. Now, if the expression of factor 8 is remaining for 5 years, I think we can just think about that this cost is higher to what we are expecting. And also for hemophilia B, we have to consider the similar type of a strategy. And next slide is just trying to say that the cost is relatively high, and there is no doubt, and is really prohibitive for most national healthcare systems. And we do need to think about how to solve the problem, and especially for those countries that they have lack of access and have no possibility to use any kind of drugs. So the gene therapy will need the new method of procurement to assess the therapy of the patient and their healthcare providers. And next slide is also reporting one of the most important issues that has been described during this webinar, how we have to be organized in future as a hub and a spoke model, because the responsibility of hub is much higher than a spoke, but there should be an interaction because the patients should really be able to move from one center to the other one, especially if there is any side effect or any problem. Next. And the management of any side effect, as I said, should be done with a specialized hemophilia care center with a multidisciplinary team. We could not think these patients should go to the gene therapy center that they have no knowledge on hemophilia. That should be excluded because these patients have different types of the need. And the next slide is just reporting the conclusion. For me, as a physician, I have no doubt this technology of adeno-associated viruses is promising, working very well, is complex and challenging. I do need to teach to physicians and also to patients how that works. One single infusion is creating a good endogenous production, but we do not know yet for how long time this expression would remain, and long-term monitoring for the safety is mandatory. There is no doubt. And that's the reason the motivation of the patient is important. We cannot think that we are injecting this drug and the patient is disappearing, we are not seeing them. That's really not possible. And encouraging evidences on the quality of life is there. And next slide is reporting what I mentioned about the cost. And I think this is really important. The high cost can only be justified by the long-term benefit and being cost-effective compared to the conventional therapy. This is very, very important. And we have all of us to keep in our mind. And uncertainty regarding the long-term benefit sometimes makes very difficult to understand the real value, economical value of the gene therapy. But of course, this is very exciting time and we have to use it. Having the gene therapy for both hemophilia A and B in 2022 is fantastic, but we have to keep in mind all the points that I mentioned. The cost of gene therapy is difficult because it's not only the expression of factor VIII, is improving the quality of life, is the reduction of the orthopedic surgery, bleeding management and hospitalization, and everything together. And that type of cost management analysis is not there yet. And the gene therapy should be understood and explained well to all suitable patients. And final decision should be done. I want to really, really stress this point with physician and patients. And the patient should be motivated and should understood exactly what that means, because otherwise I don't think any patient should be enrolled and be treated with a gene therapy. And this was what more or less was in my mind, and I'm pretty sure there would be more and more with my colleagues and who is participating in this meeting on virtual call. Thank you.
Thank you very much, Flora, for this excellent presentation. Also the clear statement in pointing out also the economic value of gene therapy, which is really very much important point beside of the discussion about efficacy and safety in the hub and spoke structure. Thank you to all presenters for your excellent presentations and highlighting gene therapy and the hub and spoke model from all the different aspects. And now we have 10 minutes to that I may ask you some questions from the audience. And I would like to start with one question to you, Dan. So you pointed out that there would be some discrepancies between the different factor 8 and factor 9 levels, so chromogenic and one stage. And is there any evidence which test would be more clearly associated to the clinical phenotype after gene therapy? And the second question, do you think we should always measure both tests in the future for patients after gene therapy?
Great question, Wolfgang, to which I don't have an absolutely clear answer. I mean, I think we have examples in non-severe haemophilia of discrepant mutations where, depending on whether one stage or a chromogenic is utilized, can either have somewhere in the kind of mild haemophilia range or indeed normal by the alternate one, and there's reverse discrepancy as well. What I find taking those patients through surgery is I use the one I'm most worried about. So we had a guy having very significant heart surgery recently, and so one could make a case for maybe the higher one being normal by chromogenic, for example, and say, well, you don't need to actually give any supportive treatment. But actually, because that's an academic debate, arguably, we chose to support the lower figure. And I think similarly at the overexpression level, particularly for 9 versus 8, given 8 is an acute phase reactant and doesn't seem to have the same durability over long term that gene therapy at the moment, overexpression of 9, I think we need to know whether there's discrepancy between those results and which number is being reflected to us in the publications. So I think seeing both in data coming through clinical trials, I think is really important. I think the ongoing debate about then which one do we worry about or which one do we use to calculate our dosing for trauma or surgery or bleeds, I think will be a kind of context specific issue.
Yes, thank you so much, Dan. There is one question for Robert. So Robert, you highlighted the motivation and also the commitment of the patient. Can you please explain a little bit more what kind of motivation should be expected in patients who are interested in gene therapy?
Yeah, we mentioned already the follow-up, Wolfgang, that it's very hard in the first 6 months, every week, blood draws for expression, for liver enzymes. And of course, you need a motivation of the patient to go through gene therapy and to have a certain commitment. I think it's very difficult from my point of view. At the moment, most of us think if the patient is very adherent to the visits in the center and with the prophylactic regimens, then he will be adherent with the requirements of gene therapy. My impression is that some patients who were not very adherent to their former therapies, they gain a lot more from gene therapy and they might have a higher motivation. So it's quite difficult to foresee this. And then I think it's the value of the hub and spoke model, because the spoke model knows the patient for a long time and they can assess more clearly, in contrast to the hub center, how motivated the patient is, how adherent the patient is, and what will be his commitment. Yeah, and so it's, from my point of view, the important role of both centers in patient selection.
Thank you, Robert. There is one question in the chat. I think it's to all, but I would like to ask Declan first. So the question is there, in your opinion, obviously in adults, a right time to perform gene therapy? Or maybe if we ask the same question in a different way, would there be a time which is not best for performing gene therapy?
So I think from a systematic perspective, and Flora highlighted the economic elements of it, is we have to ensure that it's an option in the first place. And I think that'll be a big step as an alternative option. So it's not we are switching or we are giving you another therapy, which has pretty much been the conversations in the past. This is, you're on your current therapy and this is an option for you going forward. I think if with that in mind, what we have is the opportunity for an individual to make that decision. So whether it's early on in your 20s, in your 40s, in your 50s, 60s, it's a very different thought process for each person. So, and it's highly dependent on the individual's circumstances. And that's why that conversation between the clinician and patient has to be so structured to make sure that somebody moving forward with the decision isn't doing it for a reason that may not be associated with the results that they might get from gene therapy. And that's something that we do need to be cognizant of. But I don't personally think there's a right time.
Thank you so much, Declan. I think it really depends on the individual situation of the patient. And the age of the patient can play a role, but this can be seen very differently. And there is one very interesting question from Cedric Hermans. I would like to address this question to Flora. So should a gene therapy of hemophilia A and hemophilia B be considered separately because there are some different modalities, the need for immunosuppression, and also the factor correction would differ between these both indications?
I think there is no doubt that results of hemophilia A and B gene therapy are different, but we have to consider also some of the bias related to the type of the design of the clinical trial, because the 80–90% of the ALT increase that we are seeing, I was just looking on the data yesterday, is interesting. Only, I think, about 10% they have the severe ALT, more than 3 times more. So now the question is whether we have to consider all those increase of ALT should be treated with the steroids. So that's the reason I think we have to pay attention about the data analysis that we are doing. So factor 9 expression is remaining longer, there is no doubt, at least for the experience that we have, is more stable. So the durability is much more and also is more stable. But again, we have to see whether this is valid for all clinical trial or only the result of one single clinical trial we are seeing. And in terms of the ALT increase, we definitely need, as a group, as a scientist, besides with the hepatologist, when is the time to be worried about the increase of ALT? So at the end, I think definitely we have to consider them separately, but we have also to consider the result that we have achieved for factor 8 is not really low. We had the normal level of expression for almost 3 years, and this is really something we would not have imagined a few years ago. I think we do not have to underestimate this result.
Yes, I agree. So these results are excellent and could not be expected the last years ago. And I think we have time for another additional question, and this is quite similar. I would like to address this question to Dan. Are there any trials on hemophilia A that are expected to achieve longer expression results of factor 8 activity levels than the 5 years which now have been reported?
Wouldn't it be wonderful if I had that crystal ball? I think the things to watch, so the answer is I don't know. I think we need to watch how data is presented. I think we need to not overinterpret early data. And I think seeing data mature through the kind of 3 through 5 years, that will be the only way we understand durability innate. I think understanding whether different technologies are able to achieve the same expression levels, and is there a compromise between an expression level and a durability? If you get a lower expression level, might you put the hepatocytes under less stress, which actually gives you greater durability? I think that's been mooted. I think that's not yet confidently proven, but these are the kind of issues we need to be watching out for over the coming few years.
So thank you very much. And maybe I can add that the story of gene therapy of hemophilia A is not at the end. So we are expecting new results also from the biomarine study, because these patients are followed for a longer time. And it would be interesting to see how long the expression of factor 8 would go on and continue, and how long patient may benefit from this. So with this, I would like to thank you very much for these great presentations and all the issues you highlighted regarding gene therapy and hub-and-spoke model. I would like to address one final question in the chat. I think this also would be a potential treatment for the developing countries. There are some pros and cons, but if there would be some kind of structure, it could be imagined that patients with severe hemophilia could benefit from this one single administration of gene therapy construct in the future. But of course, we are not at this stage yet. And with this, I would like to thank the speakers once more for this great presentation. I would like to thank you for your attention. And finally, I would like to thank Angelos and Pascal from EAHAD for preparing this event and doing all the important work. And please give us your feedback with this QR code. It's only a 2-minute survey, and it will help us maybe to organize some other webinars in the future. So thank you all for this very interesting webinar, and hope to see you soon. Have a nice day.